Yes, we hear and see you perfectly.
Okay, that's great. Yeah, so my lab at Cold Spring Harbor Lab is interested in understanding and controlling neural activities from the perspectives of neurotransmission centered around NMDA receptors, and then also from the perspectives of neuromodulation and inflammation, mainly mediated by large-pore channels. And dysfunction of these ion channels are implicated in neurological disorders and diseases, including Alzheimer's disease, depression, and schizophrenia. So we're interested in not only gain some understanding of these channels, but also developing the compound and reagents against them to control neuronal activities. And we're implementing structural biology, including cryo-EM, X-ray crystallography, and then we couple that with electrophysiology. As Marcus indicated, today we're going to talk about large-pore channels. Okay, now we're stuck. Can't move my slide. So I'm going to try again. Okay, great. So the definition of large-pore channel is the type of ion channels or type of channel that can not only mediate permeability of ions, but also large metabolites such as ATP. And these are controlled by stimuli such as proteolysis, extracellular calcium, and membrane voltages. And these are all involved in various biological processes such as phagocytosis, taste sensation, neuroexcitability control, and gap junctioning. And the bona fide and the most classical large-pore channels are connexing gap junctions. It was discovered as early as 1960s by cell biologists. For example, shown here is the multinuclear cell synapses, where you could actually see the gap junctionings in this negatively stained electron micrograph. And in the 1980s, Unwin basically just came up with a projection map of the gap junction crystalline, where he was able to understand that the gap junction is indeed a dimer of hexamer assembly. So later on, cryo-electron crystallography, X-ray crystallography, and most recently, single particle cryo-EM. And the resolution can actually be attained as high as 1.9 angstrom by Stephen Rachel's group, which is a remarkable achievement. So that's the bona fide large-pore channels, but it turned out that there are numbers of different large-pore channels, including SWELL channel, that the second speaker is going to talk about later. After my talk, connexing, just talked about, connexing is the invertebrate connexing or invertebrate gap junction. And most recently, Pannexin and calcium homeostasis modulator, or I'm going to call it CALHM. So we're going to basically just focus on CALHM and Pannexin. And most importantly, there's absolutely no sequence conservation amongst these large-pore channels. So they're actually different entities, but there's some structural similarity. So what about CALHM and Pannexin? So CALHM signaling modulates the neuroplasticity and amyloid beta accumulation, and consistently the point mutation, proline 8060 leucine mutations in CALHM1 was implicated as a risk factor for late onset Alzheimer's disease. And later it was shown that it is involved in taste sensation, lung airway, beat frequency control, and it's also been implicated in depression pathway. Pannexin signaling is involved in inflammatory response and the thymine signal for phagocytosis. Some common feature between Pannexin and CALHM is that they don't form any gap junctions, so they're non-gap junctional channels, and they're also voltage sensitive and known to permeate both ions and ATP. So what it means is that the pore size has to be larger than 1.4 nanometer. And there's absolutely there's little or no sequence similarity between these large-pore channels. Some differences is that CALHM1 has no ion selectivity, whereas Pannexin is strictly an anion selectivity, And CALHM1 is regulated by extracellular calcium, and Pannexin is activated by proteolytic cleavage of the C-terminal tail. So let's start with CALHM. So there are 6 members. We're focusing on CALHM1 and 2. CALHM1 does show voltage-dependent current. So that's HEK patch clamp experiment, where you see some sensitivity to calcium chloride. And you could do the same for chicken CALHM1, indicating that, and then see some current indicating that there's a functional conservation throughout species. And most importantly, it's also blocked by ruthenium red as well. And if we do the same for human CALHM2, we see little or no current. At least in our hand, we don't actually see that. So we think that human CALHM2 is non-functional, or it doesn't actually form an ion channel. In terms of sequence or topology, it's been predicted to form or have four transmembrane helices. CALHM1 has this LDS signature sequence, aspartate 120, critical for ion conductance and calcium sensitivity, as indicated by the Kevin Foskett's group at UPenn. And then the cytoplasmic domain is more variable than the transmembrane domain. So some questions about CALHM2 or CALHM1. So what is the overall structural fold? Is it similar to the rest of the large-pore family? How do subunits assemble to form the actual ion channel? Is aspartate 120 forming the actual pore? And what controls its function? Why is CALHM1 conducting and not CALHM2, 4, 5, or 6? So why is CALHM1 the only conducting channel? And where are the Alzheimer's mutations? For example, proline 86. So those are the fundamental questions. Some technical notes. So we expressed everything recombinantly using our kind of novel expression system called early back that we developed in our lab. So we expressed them, purified them, and reconstituted everything into lipid nanodisk and conducted single particle cryo-EM, not X-ray crystallography in this case. So shown here is the electron micrograph, 2D classes, and the 3D reconstruction of the structures. And shown here is the first structure of CALHM1, chicken CALHM1, in the absence of calcium. So we think it's in the active form. The resolution of the structure is 3.5 angstrom, but within the transmembrane, the local resolution is about 2.9 angstrom. So where you could actually see the clear electron density, even water molecules, where you could actually reliably build a model for CALHM molecules. It's an octamer, 8-mer, and there's an extensive interaction between the CTH, the cytoplasmic helices, which determines the oligomeric state. With the first class, you realize that CALHM1 has absolutely no structural similarity with the remainder of the large-pore channel family members. So that puts CALHM1 a structural outlier within the large-pore channel family member. And why is that? Let's look into the monomeric structure of CALHM1. So there are 4 transmembrane helices and a CTH, which is a signature sequence and signature helix for CALHM family members. So when you view transmembrane helices from the top of the extracellular side, you realize that for connexing, connexing, and BRAC channels, the volume channels, you could see that the transmembrane helices are oriented in a clockwise manner, all of them. Whereas if you try to do the same for CALHM1, you would see an S shape, indicating that there's absolutely no conservation of the helical orientations within CALHM1. And this is probably one of the reasons why CALHM1 is an outlier structurally from the rest of the large-pore family members. Just a quick note about the structure. We're actually missing about 20 amino acid residues. We actually ended up not building it, but I just wanted to also mention that there's actually a density in the middle of the pore, likely indicating the N-terminal residues, and arbitrarily measure the diameter of this pore. The diameter of this pore is about 20 angstrom, indicating that this is large enough for ATP permeation, and it is consistent with our prediction that the structure represents the open channel, open active channel. Where are the critical residues? Let's start with aspartate 120, which is critical for calcium sensitivity and the ion permeation. So aspartate 120 is not forming the actual pore. It's facing the pore, but it's actually important for intersubunit interactions. Not critically involved in the permeation itself. What about proline 86? Proline 86 is actually far away from the actual pore. and it is located in the cytoplasmic tail, probably just mediating a signal transduction for A beta accumulation. Let's look into the structure even more in more detail. So there's an extensive interaction between TMD4 and TMD2. So they're mainly hydrophobic interaction, and that's about the only interaction within the transmembrane domain. And the major interaction is within the cytoplasm between CTHs. And you could see the series of hydrophilic and hydrophobic interactions between these two helices, CTHs, and this is determined by the angle between TMD4 and CTH, and this angle is determined by the position of this proline residue within this loop, which is important for defining the oligomeric state. I'll get to that later. So the next question is, why is CALHM2 not conducting? CALHM1 is conducting, but not CALHM2. To answer this question, we step right into cryo-EM and then solve the structure of human CALHM2 at about 3.6 angstrom overall resolution. Turned out that CALHM2 has 11 meric assembly, not octameric assembly. So this is an interesting case where the same family members, CALHM family members, have different oligomeric assembly. So it's 11 meric, and arginine 124 is at the equivalent site to aspartate 120, and it's also mediating the intra-subunit interaction facing the pore, and then the major transmembrane helices interactions are between TMD2 and 4 in a similar way to CALHM1 interaction, but the major difference is the mode of interaction between the CTHs in the cytoplasm. So there's no sequence conservation within the CTH between CALHM1 and 2, and then they involve different residues and different interactions. And this is mediated again by the angle between TMD4 and CTH, which is regulated by the positioning of this proline, which is different from the CALHM1. So if you actually look at the superposition, you could easily see that TMD4 CTH angles are different, and they're again controlled by different positioning of proline residues in black. Let's look into the pore regions. The CALHM1 has extremely hydrophilic pore, and that's not surprising because they're conducting channels, whereas human CALHM2 has more hydrophobic pore regions. Okay, so this indicates that it is probably rather unfavorable to permeate hydrophilic compounds such as ATP or even ions. What we ended up observing in cryo-EM is the amorphous density in the middle, which we don't actually see in CALHM1. And we speculated that we probably have some kind of lipid density within the CALHM2 pore. So to validate that, we conducted MD simulation in collaboration with Stephen Tucker and Charlene Rao, his student. So what she was able to do is to reconstitute CALHM1 and CALHM2 into POPC lipid bilayer. And then she was able to conduct coarse-grained molecular dynamics, where she would see an unstable placement of lipid within the CALHM1 pore, but stable placement of lipids within CALHM2 pore, indicating that CALHM2 can accommodate lipid into its pore, and then most likely the density we saw in cryo-EM represents the lipid. So you can imagine CALHM2 to be plugged by lipids. So this is probably one of the reasons why CALHM2 does not have any conducting activity or channel activity. Let's compare CALHM1 and 2 monomers. So these are the monomeric structure of CALHM1 and 2 in cyan and magenta. You could superimpose them and see that the transmembrane structures are quite similar to each other, and the extracellular domain structure is quite similar to each other. The only difference is the angle between the TMD4 and CTH, defined by the number of proline residues at a different position. So we speculated that it's this angle D4 and CTH that defines the oligomeric state. So we wondered, to validate that, we made a chimeric proteins where we have a transmembrane from CALHM1 and the cytoplasmic domain from CALHM2. So what happens to the oligomeric state as a result of doing that? So to do that, we conducted single particle analysis again, this time on chimera of CALHM1 and CALHM2 in cyan and magenta respectively. And as you can see, the swapping of CTH ended up converting CALHM1 channel from 8 MER to 11 MER, okay? Just like CALHM2, indicating that the cytoplasmic domain is important for controlling the oligomeric state. So we see similar interaction in TMD2 and 4, indicating that this TMD interaction is quite flexible. But what we now end up seeing is the specific interaction at the CTH represented by the CALHM2 sequence, which basically just defines it as the 11 meric channels. And the consequence of converting CALHM1 into 11 meric channel is the null functionality. So this is the recording for the bona fide CALHM1. If we do the same but convert it to 11 MER, we end up losing the function, indicating that you really have to have 8 MER and no lipids within this pore to be conducted, to have a conducting channel. It doesn't show in this figure, but I just also wanted to mention that by converting CALHM1 into 11 MER, we actually ended up observing amorphous density in the middle. So it's as if we actually induce the lipid filling by converting CALHM1 channel to 11 MER. All right, so in a nutshell, we think that the CALHM1 is 8 MER and CALHM2 is 11 MER. And when the pore is large enough, like CALHM2, the pore can be plugged by lipids. Okay? And as we saw, we just convert the 8 MER into 11 MER, we end up seeing the same thing, lipid plugging. And the field now has a different CALHM structures, including 4, 5, and 6, and they happen to have large pore channels, starting from 10 MER to even 13 MER, and they all happen to have lipid plugging. Same as CALHM2, and the common denominator is that they all don't have any ion channel activity. So we think that this lipid plugging mechanism is something that's common in CALHM family. So what it does is actually remains to be a question at the moment. Okay, so using the remainder of my time, I'd like to talk about Pannexin channel. So this work was done in collaboration with Kawate's lab at Cornell University. So there are 3 members of Pannexin families, Pannexin 1, 1, 2, and 3, and we focus on Pannexin 1, which is the most well-studied Pannexin subunit. They're supposed to have 4 transmembrane helices, and it is activated by caspase cleavage at the C-terminal domain. Shown here is the electrophysiology of the human Pannexin 1, which is blocked by a compound CBX, it's a channel blocker. And then we've done the same for frog Pannexin 1, which basically just happened to be the subject to our structural biology. So they can also be blocked by CBX. And then we also designed a C-terminal truncation, which represents the active form of Pannexin 1, and it is still sensitive to CBX. Okay, so this is what we focused on. So we basically try to capture the active state of Pannexin 1 in this particular case, implemented cryo-EM in the same way, and shown here is the first structure of Pannexin 1 that we published about 2 years ago at about 3 angstrom overall resolution. Again, we can clearly see the transmembrane densities, which basically allowed us to model everything within the transmembrane. It is the heptameric structure rather than hexameric structure. Hexameric simply was predicted, but it turned out to be heptameric. And then at the extracellular side, there's an interesting motif, clusters, essentially clusters of tryptophan residues and the arginine residues, which is situated in a suitable position to form cation-pi interaction. So you can actually view Pannexin to have a highly positively charged at the extracellular domain. Just remember, Pannexin channels are chloride sensitive channel. Okay, I'm just showing the monomeric channel. Just wanted to show that the Pannexin channels are quite similar to the rest of the large-pore channel family, but not CALHM channels, indicating that CALHM channels are outlier within the large-pore channel family, at least in the context of structural biology. Pannexin actually happen to have the closest structural homology to Nexin 6. Just also wanted to let you know that we do have a CBX bound structure. So we've done everything in the presence of lipid nanodisk. So what I showed initially was the apo state and the active state, and this is the block state. So CBX basically recognizes this extracellular motif at the extracellular domain through hydrophobic interactions. There are numbers of Pannexin structures that are available, and for those which was solved in the presence of detergent called digitonin, happen to have a density in the middle, despite the fact that they claim it as active and open channel structure. That's because digitonin or detergents such as DDX happen to have a cholesterol motif. So I actually propose that these structures represent digitonin blocked Pannexin structures. But nevertheless, with or without CBX, you would see the same diameter of the pore, so there's really no changes locally. And the size of the pore, whether there's a presence of CBX or not, happen to be about 9 angstrom or so, which is large enough for chloride conductance, but not ATP conductance, indicating that for ATP to permeate, there has to be something more. The ion channel has to probably just dilate even further. So what about this extracellular motif? Is that actually responsible for chloride selectivity? The answer is yes. To validate that, we've done some electrophysiology experiment. So what Eric was able to do was to basically just measure IV curve in the presence of sodium chloride, sodium gluconate, and then NMDG chloride. Okay, so done that, and then basically show that there is a chloride sensitivity. So you would actually see a rightward shift when you do the recording in the presence of sodium gluconate. So is charge important? The answer is yes. So when you site direct mutate arginine 75 to lysine, but with a conserved overall positive charge, then you would actually not see any differences, indicating that it's actually the charge that's critical for chloride selectivity. What happens when you get rid of this charge, right? You would lose chloride selectivity. That's not surprising, but surprisingly, when you change that residue to glutamate now to mediate some sort of anion pi interaction, then you would not only lose chloride selectivity, but you would actually also gain sodium selectivity. Again, indicating that it's actually the charge around this tryptophan 74 that basically just creates the overall charge, and then that really determines the selectivity or anion or cation selectivity. So again, it's the charge that's mediated by the backside of these tryptophan residues. So our tentative conclusion about large-pore channel paradigm, so there has been lots of mystery. We solved some of them, and we basically just realized that the Pannexin architecture is similar to other large-pore channel members, despite the fact it's heptameric, unprecedented. The monomeric structures are actually quite similar to each other, whereas CALHMs are outliers and they have a bunch of different oligomeric states, so it's a weird channel. Pannexin has a narrow cation pi constriction for chloride selectivity, whereas and the current structure indicates that it's open, and then the chloride permeability is regulated by the charge. And the pore of CALHM1 is wide enough to permeate ATP, whereas CALHM2, we propose that is plugged by the lipids, okay? And so is CALHM4, 5, and 6. So it really remains to be seen if whether or not CALHM2, 4, 5, and 6 actually have the ion channel activity. If so, there's got to be a mechanism to remove the lipids from the pore regions. So that remains to be seen, and that's what we're working on at the moment. So with acknowledge the people who's done the work, the majority of the work was done by my postdoc, Johanna Sirianen, who's actually looking for a job right now. So if any of you are looking at watching this seminar, please, she's fantastic. And also, Kevin Michalski was involved in Pannexin project. I'd like to thank the supporting staff, collaborator, Shaolin, Stephen Tucker, Toshi Kawate, and Eric. And then this work was funded by RF Fund, NINDS, and others. Thank you.
Thank you, Hiro. That was fantastic. So yeah, it is my understanding that we keep the questions for the end. Is that okay if you stick around then for the next, and then we do the questions around 9:50 or so? So yeah, and so it's a pleasure for me to introduce our second speaker, Zhaozhu Qiu, who virtually visits us from Johns Hopkins, where he's an associate professor in the Department of Physiology and Neuroscience. And yeah, I mean, his lab was really at the forefront in cloning and identifying several new chloride channels that he will be telling us about now. So thanks, Zhaozhu, for joining, and looking forward to your talk.
Thank you very much, Marcus, for the very nice introduction, and organizers for inviting me to give the talk. Can you hear me okay?
Yes, we hear and see you well. Great.
Great, thank you. So my lab, as Marcus just mentioned, that we are interested in chloride channels. Actually, chloride is the only major negative charge ions, and most of people study potassium channels, sodium channels, and calcium channels. You know, for good reasons. They are underlying action potential, signal transduction, but you know, few people study chloride channels, but think they're also very important. Let's see. So chloride channels actually play very important roles, and chloride concentration are regulated inside of cells. For example, in neurons, typically they express a lot of transporters will extrude chloride, so makes the chloride concentration inside of the neurons very low. In that case, activation of ligand-gated chloride channels such as GABA receptors, it will be inhibitory. However, in most of the cells, like the epithelial cells, where they express various kinds of transporters, NKCC, for example, load chloride. So the chloride concentration inside of cells are actually relatively high. And in those cases, when the chloride channels in apical membrane open, the chloride goes out. That underlines the fluid secretion, for example, CFTR and calcium activated chloride channels. In reality, there's actually many kinds of chloride channels hasn't been cloned. For example, when we started this, there's a volume regulated chloride channel. I will talk about a little bit in the end of the talk, which is one of the large-pore channel Hiro just talked about. There's also channels activated by acid, and collectively these channels have various roles such as fluid secretion, regulating excitability, and regulating pH of the organelles. So I was getting back to that. So one of the reasons that the many chloride channels hasn't been cloned, meaning that you can see the ion channel activity by patch clamping, but you don't know which gene it is encoding them. It's because various families of chloride channels just talked about, they don't share any sequence homology. This is quite different from most of the cation channels, such as voltage-gated sodium channels, potassium channels, and trip channels. They all have sequence homology of the voltage sensor domains or pore domains. So by looking at sequence, you know which gene encoding these channels. So that's not the case for chloride channel, and that cause a problem where we don't know the molecular identity of many of these channels. So in my lab, we identified two families of these channels. One is activated by proton or acid. Another family is activated by cell swelling. So today I will mainly focus on the proton activated chloride channel. And acid, obviously very important physiology in disease. And in many disease states, there's acidosis happening, causing tissue damage and pain. And also many intracellular organelles also acidic, and that's important for many of its function. And one of the receptors activated by acid is this chloride channel, as shown here. If you submit a whole cell patch clamp, and when you perfuse acid, you see this large outward current and activate in a relatively low pH situation. And you could imagine that this might be play a role in acidosis or disease. But one thing interesting about this current is that they actually widely expressed, even in 293 cells or CHO cells or many cells, which people recorded, and suggesting there might have some physiological function of this current. And there's two reasons why this current didn't call too much attention. One of them is that it's auto-rectified. So these are the positive voltages. So if in the negative voltage you hold the cells, you don't see too much of the current. And second, again, the sensitivity to the pH is relatively in the acidic side, although this is modulated by temperature. So at the 37 degree, the channel are more sensitive to the acid. And even though the physiological relevance of the channel is not clear when we first started this, we thought that, just maybe clone this and we try to figure out what it's doing later on. So that's what we did. And there's a number of, people tried to clone those channels, and there's some challenges, as I discussed in the beginning, that if you just look at known chloride channel families, but they're not conserved, so that's not successful. Also, these channels generally chloride channel lacks good biochemical probes like toxins or ligand, which you can do by chemical purification. In addition, that these cells express a lot of endogenous chloride channel activity. So when you do cDNA expression cloning, also tends to be not very effective. So after we kind of read the literature, we thought maybe we should do a loss of function screen and using siRNA to knock down gene one by one and see which gene would be responsible for this channel activity. And in order to do that, we go into a very robust chloride channel assay, which is called a YP quenching assay. Essentially, this is a fluorescent protein, which once it's binded to anion, particularly iodide, the fluorescent will quench. And we reason that if we make a stable cells express this YP, and perfuse cells with acid, so the channel would be open, and then we're adding iodide in the solution because there's no iodide inside of the cell, and also chloride channels are not selective because they don't have to, because chloride is the only anion in a human system. So when you're adding iodide, the iodide can also permeable through the channel, they get into the cell and get it quenching. And that's exactly what we found. So here's the fluorescent traces when you add acid, which activates the channel, and when you add iodide goes through the channel into the cell, and you get a quenching, and this quenching is dependent on low pH. So you get a very nice dose-dependent curve, and we're ready to go. So we're lucky to get a hand on the siRNA library, which targets all two transmembrane proteins. We figured that we try to look at the channel, and channel has at least two transmembrane, and that actually has finite number of genes. There's about less than 3,000 of such genes, and a third of them are GPCR. So, but anyway, we screen all of them, and this is a typical plates. You have a 384 word plates, and each way you have cells, you have one siRNA being transfected, and in most of the cases where you get big quenching because the channel is intact, the iodide get into the cell and they quench, but you occasionally have wells where you have less quenching, and that would be a potential hit. So we ran through the library. So it's about 3,000 genes and 4 siRNA per gene. So it's about 12,000 siRNAs wells. And we're very excited to find the top. We had one best hit, which has 3 shRNA are very potent. And this basically is the top hit, and all the others only have one siRNA working, it's just false positive. And even better, this is an unknown protein. When we identify, there's no papers on this gene. It's called TMEM206. Basically, it's just a number. So we call them PAC for proton activate chloride channel. And before we get too excited, obviously we want to do recording to show that this gene indeed is necessary for the channel activity, which it is. And also, when you overexpress, you get more channel activity. So it's also sufficient. And it has 2 transmembrane. And my postdoc Junhua and student James did all mutagenesis, and we found that TM2 are particularly important for iron selectivity. So we think this is this protein likely to be the channel itself. And we also started to do biochemistry to figure out, is the protein has monomeric structures, and this accumulated with a collaboration with the Lu and Du lab at the Van Ender Institute with a talented postdoc Zhen, who we purify the protein and determine the chloride structure first at the neutral pH. And obviously, this to us is it's kind of eye-opening, because previously, from cloning the channel to solve structure maybe take 10-20 years, as Hiro was illustrating the history of the Pannexin channels and all these structures. But now the advance of cryo-EM really made it possible, basically in months, that you would get a structure. And obviously, we are very, very happy. It's very exciting, because it is a channel, right? It's a trimeric structure. It has a large extracellular domain. Presumably, that's the pH sensing domain, and it forms a channel. So that's very reassuring. Fantastic. We went on to determine the structure of not only at neutral pH but also at the acidic pH, as shown in this movie here, that to see how the pH would induce a conformation change. So this is pH 8. You have this two transmembrane domains, a trimeric structure, and we found there's particularly histidine residue, which upon protonation, presumably will switching the conformation and interacting with a cluster of negative charge residues, which is called an acidic pocket, and this at least partially be a component of the pH-dependent regulation of the channel. Actually, we made mutations of these histidine residues and also the acidic pocket residues, and all of them changes the pH sensitivity makes the pad more sensitive to pH. So it's a remarkable machinery to sensing the pH extracellularly and change conformation and conducts chloride. So that's all great. And now back to the question of physiology, why you would have the channel. And obviously, the first thing we think about is maybe it has to do something with acidosis and disease. And indeed, people are postulating that in ischemic stroke, for example, when you have blood clog, you have increased lactate production and have acidic environment, and these channels would be open, and that chloride get into the neurons, and you get swelling and get cell death. And we set to test this hypothesis. First, we actually made a pack knockout mice, and those mice are viable and generally okay. So we was able to prepare the neurons from these mice. So it's a knockout neurons, and we treat those neurons with acidic solutions. And indeed, we found that those acidic solutions would induce neuronal cell death, as indicated in the red. However, in the knockout neurons, and these deaths are reduced. So it appears that PAC actually is partially contributing to the acid-induced cell death, presumably by inducing the chloride influx of the cells. So what about the animals? So we turned into this ischemic stroke model, which is called mid-cerebral artery occlusion model. Essentially, you basically do a surgery and put a filament into the brain of the mice and blocks the mid-cerebral artery. And 24 hours later, you cut the brain slices and do a staining. So the red part, the red is the live tissue, and the white tissue is the dead tissue. So that's a very basically cell death assay, but it's kind of in vivo setting. So we did that in the knockout mice, and we found very excitingly that the reduction in the total infect volume of the white volume of these knockouts, and particularly in an area where it's not in the core of the ischemia and in the periphery tissue, where the tissue still can be salvageable, and also that correlating with the improvement in the neurological score. So this data suggests that PAC at least is one component in the ischemic stroke, well, in the acidosis settings, the contributing to cell death. So we're very excited to screen in small molecules to modulate it in the future and see whether those can be beneficial in the in these animals. So so the next question obviously is that the PAC channel is, as I mentioned, is widely expressed, but it's it's not for a disease setting where it's causing damage. Obviously, it might have some more housekeeping or basic physiological function. So what it is. So that's the importance of cloning the gene would make a difference, because now we can look at where they are localized. And this basically is a kind of a eureka moment when we transfecting PAC with a flag tag. We found that although it has some faint expression in the cell surface, however, predominantly the protein actually localized intracellularly. And this immediately stimulates our curiosity, that suggesting that channel may be in the acidic compartments of cells. So indeed, we look at various markers, for example, EA1, which labels early endosome, and transferrin, which is the recycling endosome, and they have very nice colocalization. To our little surprise, that LAMP1, which is lysosome, they actually do not co-localize. So we're getting back to that later. So it appears that PAC is in the cell surface, because we know if you do patch clamping, you get whole cell currents. But if you do immunostaining, they actually mainly intracellular. And to exclude that it's not just artifact, we transfected these plasmids and they just end up in endosome, lysosome. We actually raise the antibody to PAC, as shown here. It's specific, it's a wild-type cell, it's a knockout, so you don't have a band. And in these settings, if you do immunostaining, again, it's still intracellular endosome, so suggesting that it's endogenously itself. So we look at the PAC sequences. Actually, the N-termin and C-termin have very short tails, and we did a sequence alignment, and we found there's one motif, the tyrosine motif, YXXL motif, which is a classic motif mediating AP2 complex binding and the class 3 mediated endocytosis. As expected, when we mutate in this, either the tyrosine or the leucine, that in both cases that the protein will be mainly stuck in the plus membrane, suggesting that the endocytosis is an active process regulating localization. And obviously there's a big literature on how chloride be important in endosome, for example. So how this works is that the endosomal acidification starts with the VATPase, which use energy to pump proton. However, the proton is positive charge. This process will go against the membrane potential increase. So how the cell deal with that is they will put chloride as a counter ion into the lumen to balance this charge and facilitates the acidification. And this was thought to be done by a transporter called CLC, which very interestingly, it actually leaks one proton and import 2 chloride. And that's how chloride get into the endosome and act as a counter ion. And there's a lot of mathematical models suggesting why this is the best solution for the endosome. But our work suggests that indeed there actually is a chloride channel. And this chloride channel, we postulated maybe sensing the pH in the lumen. So when the pH is too low, they might mix chloride. That's kind of a balanced check with the CLC transporters. So to test that, first we want to see whether PAC is really in the endosome functionally. So to do that, we turned into this whole endosome recording, as this is video provided by Dejian, as you can. So the cells are transfected with a mutant RAP5, which will enlarge were preventing them they were promoting the enlargement of the endosomes, as shown here, and they co-localize with PAC. So so then you can open up the cells and release that endosome, and then patch clamp them. So as you can show this video, where you transfect the cells with RAP5, which enlarge the endosomes. Then you have a glass pipette approaching the cells, and you start to kind of peel off, rupture the membrane of the cells, and then you kind of gently push this vesicle out. So now basically this, just like the whole cell, you can record that. Obviously, James spent a lot of time to practice in and several months and make it to work. It's a great technique that now we can have access to this endosome. So when James patched that, he again see this very nice current, actually very similar to the PAC current in the cell surface. It auto-rectifies. And this current appears to be mediated by mainly chloride leaking. So if you have low chloride in the endosome, you don't see the current, and it also depends on the pH. So if the pH is neutral pH, you also don't see the current. And to test when this is indeed mediated by PAC, we made a PAC knockout cells and we patched this, and we again didn't see the current, suggesting that PAC really mediates this acid sensitive endosome chloride currents. And we also overexpress PAC, we see more of those activities as well. So since it's already published, I will just kind of briefly summary what we think is happening. That so PAC is a cell surface and it's endocytosed by this y-axis motif, and in the endosome it seems to be function as the endosomal pH sensor, which preventing the hyperacidification. So when the pH is getting too low, again the sensor domain is inside the lumen now, just like in outside. So this was activated, it will need chloride, and this will preventing the over-acidification of the endosome. And then we found that in the knockout cells, we measure the chloride concentration, the increased chloride concentration as predicted, and also the endosome pH is more acidified. In the overexpression settings, that it will alkalize, presumably by needing more chloride. And this has implications with various receptor trafficking. So we'll look at transferrin receptor trafficking, it's being affected. So right now we're looking at many other systems where it requires the endosomal acidification for the receptor trafficking. So just as a summary of the kind of future direction of the work with the PAC, we found that actually for loss of function screen is very effective to identify molecular identities of unknown chloride channels in our hands, including PAC and also the SWELL channel we'll talk about in a minute. And PAC seems to be a potential target for disease associated with acidosis, such as ischemic stroke, cancer, or inflammation. And it has a very interesting physiological function. We think it's maybe regulating endosome pH and also endocytosis and receptor trafficking. So in the last couple of minutes, I will just quickly go through a couple of slides on another channel we cloned. Actually, the first channel we cloned with the chloride channel called a volume regulator anion channel. This is a channel being characterized for more than 30-40 years. The main idea is that the cells in our body has remarkable ability to regulate volume. So as shown here, if you put the mammalian cell in a diluted environment, the water will get into the cell by osmosis. And the cells in our body has this remarkable ability to regulate the volume back in some time, in minutes. And the idea is how it happens is that the swelling activates various channels and transporters. One of the key component is a chloride channel, where it will release chloride and also other organic osmoles. So it's a large pore channel. And because of the salt leave cells, osmosis leave cells, the water will follow. So that's how it works. Again, that the molecular identity is not known for many, many years. And this is actually the first screen we did. It's a very easy concept where instead of adding acid to activate channel, you could imagine you can just adding hypertonic solutions to activate channel and read out the quenching response. So this is secondary screen from the screen where this is scrambled siRNA. You get a lot of quenching because the channel is intact. And this is an inhibitor, like a benchmark inhibitor, and this is an assay window. And this is a secondary screen about 50 genes, and one of the gene called leucine rich repeat family containing family 8A. It's kind of a mouthful called SWELL1 because it's activated by cell swelling. It basically just like knockdown has a perfect suppression of the channel activity. And this channel type is very interesting And it's activated not only by cell swelling, but also some other stimuli such as GPC activation or apoptosis, and also intracellular stress. And this channel is also formed by a hexamer, as Hiro talked about, with other large pore channels, and it actually has this a subunit of SWELL. One is the essential subunit. We are very lucky, and there are also other subunits also necessary for make a functional channel, and combine they determine the selectivity, slightly selectivity of this channel. And this is still a very active area of research. We try to figure out how this whole system works. But for us, we are more interested in biology. It turns out this channel has a very important function in cell communication, just like the channels that Hiro just introduced. For example, people found that this channel are very important uptake of various cancer drugs. So if you knock out these genes in cancer cells, they are resistant to this cisplatin, for example, cancer therapy, primarily because the drug cannot get into the cell very efficiently. And in our lab, we focus on in the brain. So for example, glutamate and other neurotransmitters are being released from these channels and regulating synapse transmission. We also had collaboration with immunologists or virologists, where this channel can uptake CGAP, which is activated upon viral infection. And then this is a way to kind of alert surrounding cells that there's infection going on, and turn on your interferon response, and it's very cool. And we think that this also involved in release of CGAP as well. So there's some basal activity happening inside the body. It has nothing to do with cell swelling, we think. And they are involved in uptake or releasing these factors, and how this all work, and how the specificity is achieved is still kind of a big mystery. And we're kind of looking for more people to come into the field and to figure out how this works. It's very, very interesting channel. So with that, I just want to thank the people who did the work. And as I mentioned, James is a student, graduated from lab last year, and Junhua is the first postdoc who did a lot of work, and Janan did the stroke analysis, and we also have collaborators at Van Anderson. They are fantastic to solve the structure, and also the funding sources. Yeah, thank you again for your attention, and I'm happy to take any questions. Thank you.